So in today's lab, we're going to be looking at different types of media. We talked about growth of microbes a couple of weeks ago. We talked about different media that are used to grow them. Some media are pretty general, like tryptic soy agar and nutrient agar and broth that we've used are just to grow general bacteria. But what we're going to be looking at today is we are going to be looking at specific media used to grow and or select for certain type of bacteria. For example, if you wanted to specifically just look at gram-negative bacteria, then you would pick one type of agar, or if you wanted to look at gram-positive. So we know we can do gram stains for this, but this in these medias, they can grow certain bacteria, inhibit other ones. Okay, so it's kind of a starting point. With the differential media, you're able to see the difference between different types of bacteria. So things like blood agar is also called an enriched media, but it can also be differential because it can tell different hemolysis patterns. Or mannitol salt can tell the difference between a fermenter and a non-fermenter. So something like mannitol salt is selective because of the salt in it. The salt inhibits the growth of most bacteria except for the ones that live on your skin because your skin is very salty. And then the fermentation part of it, the mannitol, if it ferments, remember it turns yellow, just like the other fermentation tests that we've done. So you can actually distinguish between Staph aureus as opposed to Staph epidermidis based on the growth and the color change in the media. So in today's lab, we have lots of different media and lots of different bacteria. Okay, so I was correct in counting. We have 8 different bacteria, which are going to be in broth tubes. And then we're going to have, we have 6 plates here, 6 different plates. So what you're going to want to do is follow the directions exactly. I'm not going to make any changes in the terms of what media you're going to use and what microbes you're going to put on them, except for if you look on the cultures that you're going to use, the third one down, it says Streptococcus variant Lancefield group A, and then it says Streptococcus mitis. Well, we do not have those 2 species. Instead, we have Streptococcus pyogenes and we have Streptococcus pneumoniae. So what you're going to want to do is cross out the Streptococcus Lancefield group E and cross out Streptococcus mitis and replace those with Streptococcus pyogenes and Streptococcus pneumoniae. Okay? The rest of it is pretty self-explanatory. The one thing that you want to make sure that you do is draw the lines on the plates, but now also always make sure you draw them on the bottom of the plate because lids can turn. So say I draw a line on the top, and then I write the name of the bacteria there, and then when it gets put in the incubator, it gets changed, then you're not going to know which one it is. So always draw right on the bottom of the plate. The other thing is you always want to label on the edges, big enough to read, but small enough that it's not going to interfere with your results. Okay? So I like this lab because of the different colors. I like colors, I guess. So you have some varying red colors. So like the blood agar is a very kind of a bright red, and you can't really see through it. The mannitol salt is also red, but you can see through it. We have the McConkey, which is a little bit lighter and a pinker. And then the Eosin methylene blue is, I guess is a more of a burgundy almost color to it. And we have a couple of boring ones. They're just kind of just the regular agar color. That's going to be the salt and the phenyl ethyl alcohol. The other one is kind of a purple color. That's the crystal violet. So for this, it looks like we have 6 plates. Looks like we have 7 plates. Okay. We just count them and make sure we're correct. 1, 2, 3, 4, 5, 6, 7. Okay. So what you want to do is divide them according to the way it says to divide them in the lab manual. And then you're going to label them the way that it's written to label, keeping in mind that you're going to use the 2 Streptococcus species that I mentioned earlier rather than the ones that are in the lab manual. Okay? Now for these, when you go to inoculate them, We're going to do similar to what we did, it was when we did the milk auger and the starch auger, where instead of inoculating it by going back and forth, you're going to draw just a single line of inoculum. I don't know if you recall when we did the milk auger, we had one of the species, I think it was bacillus, that actually spread out into the other side. So by doing a small single line of inoculum, it keeps them from running together and you get the results mixed up. Okay? All of them you're going to inoculate in that manner, just one line, you're done, one line of each species. Keep in mind you're going to be putting up to 4 on one plate, so you have to be careful not to cross over from one side to the next. I'm not going to show you aseptic technique. You already need to know that. You'll be wearing gloves and goggles and doing all the appropriate safety precautions that you normally do. Keep in mind that you do have a lot of plates and you have a lot of species of bacteria, so you're going to have to be organized to make sure that you do it correctly. The one plate that's going to be a little bit different is going to be your blood auger plate. Your blood auger plate, when you put the species on the blood auger plate, after you draw your line of inoculum, you're actually going to take your loop, and you will have sterilized it, and you're just going to go across the inoculum in kind of a 45. The degree doesn't matter, you're just going across the inoculum and actually cutting down into the auger. The goal is to get a little bit of the bacteria to go down into the auger. It helps with seeing the hemolysis pattern. Okay. The other thing you want to do is make sure on these that you're careful not to cross-contaminate and also not to contaminate from your hands. I've been noticing with plates that we're getting little contaminants here and there. So when you go to inoculate, make sure that after you've done your flaming and everything, and while you're cooling, you wait. You don't open that plate until it's cool. And then once your loop is cool, you just open that plate at a 45-degree angle, make your line, and then close it. If you're doing the blood auger plate, then you're going to cut across, like I was mentioning, and you're just making a little kind of cut into the auger. Don't cut too far to where the auger is going to crack. You just kind of want to basically just kind of press down into the auger. And then, of course, as always, you're going to put the plates upside down in the incubator. These are all going to go into the 37-degree incubator. Now, on the second part of the lab, you're going to be looking for results such as growth or no growth. And so it gives you a guide as to how to write that in your results table, like a +1 being a small amount of growth and a +2 being a large amount of growth and things like that. So you're going to look for growth or no growth first. And so for things like the selective auger, that's all you're looking for. Did it grow or not grow? It should inhibit certain things. But on the differential auger, like the mannitol salt, then you're going to be looking at color changes in the media, like it might turn yellow with fermentation. The Eosin methylene blue is really cool because you know how we've been looking at the change of color in the media? But in this case, the actual culture, the actual smear of bacteria will change color. In some cases, it might turn like a metallic bluish green color. In other cases, it might be a really pretty pink color. So this will differentiate and show you differences based on the color of the bacteria change as opposed to the mannitol salt where you're looking for the change in the media. So different things you'll be looking for. So this is kind of a fun lab, just seeing a lot of different types of results.